live.
There's always a little delay on YouTube
right.
before you can push that magic button.
It's done.
Great. Let's see.
So let me check.
So how will you field how the questions handle from YouTube?
So we invite people to write their questions into the chat on YouTube, and then we usually read them out to you.
Okay.
And for the people in the live Zoom meeting, they're always invited to just unmute and ask directly if possible. All right, let's admit people here.
Maybe if you want to try to share your screen, if everything is okay. Andre?
Yeah.
Perfect.
Let's just wait one more minute to let people drop in. Hi, everyone. Nice to see you. I love it. There's always this mix of familiar faces and new, at least new names. It's calming down now with the admission. So let's make a start. Welcome everyone here live on Zoom, over on YouTube, and welcome to Andre, our speaker today. He is a computational neuroscientist who's interested in the development of novel statistical techniques for neuroimaging data. He's currently a PI at the Dundas Institute for Neuroimaging and the Dundas Institute for Brain Cognition and Behavior. His background is a bachelor in computer sciences and psychology from New Zealand. And then in 2007, he moved to the UK for a master's in neuroscience, and then subsequently did his PhD in clinical neurosciences at King's, where we met. And his topic was probabilistic machine learning methods for multivariate prediction of magnetic resonance images. And I'm actually very pleased to say that my PhD has a little thank you note to Andre, because we had a little chat on the corridor about how to improve the methods of my PhD, and it changed the entire analysis for the better. So thanks for that again. Right, without further ado, the way it works is if you have questions here on Zoom, you can write them in the chat or unmute yourself, show your face and ask directly. For those over on YouTube, feel free to write in the chat over there, and then we bring the questions directly to Andre for you. With that, welcome to the CNS talk for today. The floor is yours, Andre.
Thanks very much. It's really nice to be here. Thanks for the invitation. I'm going to talk about machine learning techniques and big data in neuroimaging, particularly with a focus on clinical neuroimaging. I, in fact, changed the topic of my talk from what was advertised, so I'll explain that in a moment. But just to start with, I would like to start with something like a mission statement, if you like, that this is what we are all about. In our lab, we aim to develop innovative machine learning techniques, apply them to big data, and aiming ultimately to have impact on problems that matter in psychiatry and neurology. So the plan for today's talk, I'm going to talk about 3 topics. So I'm going to talk about using big data cohorts to make accurate predictions at the level of the individual participant. That was actually the advertised topic of the talk. But I'd also like to focus a little bit on some of the newer things we've been working on recently. So learning techniques for learning neural representations, and also at the end, just a little bit about methods for integrating different types of technology to get a holistic perspective on each individual, ultimately aiming to get a more accurate predictions or stratifications of individuals. So let's start with this topic, and I'm going to motivate it by the notion of using biological measures, for example, neuroimaging, but also things like genetics, in order to make decisions about individual people. Now, in psychiatry, you'll be familiar with this notion, because, for example, blood tests are used in this fashion to determine which kind of diagnosis you might have, or what treatment allocation would be optimal for a given individual. And then, of course, in psychiatry, we're handicapped because we don't have those sorts of biological markers. So there's been a search for a number of years for techniques and measures derived from, for example, neuroimaging that we can use for this purpose. This deviates a little bit from the classical notions of things like statistical parametric mapping, GWAS, where we are aiming to determine a difference between groups. So on average, do patients differ from controls in this particular measure? For example, using a t-test or something like that. Now, the problem with this is that from a decision-making point of view, clinicians need to treat individuals, not groups. So this doesn't really get us very far beyond the idea of mapping of potential mechanisms. So what we started doing a number of years ago was trying to train classifiers on the basis of patterns of activity, for example, that aim to separate these separate classes, nominally patients and controls, or different diagnoses from one another, on the basis of the pattern within the data. And by applying this to a new data point, this learned decision function that we've learned from a given data set, we can make predictions at the level of the individual. So we've moved from group level inferences to subject level inferences. And we can also use this method by looking at the accuracy, for example, as a way to quantify the separation between groups. So how different are groups from one another on the basis of the pattern that we observe. And this was this is a very old study, but this was a study that was one of the first ones I worked on, in fact, and we used this notion to separate, we used classifiers that were trained on fMRI data acquired during an effective processing task. A support vector machine was a classifier that we used back in those days. And we were able to discriminate both patients from controls, but also to predict which individuals would respond to a particular treatment or not. So this has, there are many studies in this direction. I'll refer you to this, to some of these reviews. One by a PhD student, a former PhD student of mine, Thomas Wolfers. They've been applied to effectively any brain disorder that you could name, and predicting outcome, diagnosis, you name it. The problem is, so if we sort of dig into the literature a little bit, and this was something that Thomas did during his PhD, the picture becomes a little bit more sobering. And so what all of these things are plotting is the accuracy obtained by these models as a function of sample size here on a logarithmic scale. Now you can see that paradoxically, as we increase the sample size, the accuracy goes down, which is not what it should do in machine learning terms. So we should be able to explain better the pattern that differentiates between individuals as we increase the sample size. There are clear vibration effects. That means all these small samples where there's a lot of variance in the accuracies, which is really, and so this is all really quite bad news for the field, I would say. And I attribute a lot of this to heterogeneity within these clinical cohorts, which we're not doing a very good job of modeling. And it's not difficult in clinical cohorts to think about where this heterogeneity might come from. So we can think of things like multiple diagnoses in the same individual, comorbidity. We can think about different symptom presentations in different individuals. So one person with depression may have a different symptom profile from another. And we can also think about heterogeneity at the level of mechanisms. So different biological mechanisms converging on the same clinical phenotype. And so all of these can contribute to what we observe in a clinical sample, and all of these things will limit our ability to separate, to discriminate these conditions. And in many ways, this preempts a paper that was, this is a theme that emerges in a paper that was recently published in Nature. And particularly drawn attention to Pierre Bellec's comment on this on Twitter about this paper. And really, the way I look at it, I mean, I would very much agree with Pierre in this conclusion that really we're not doing a very good job at all at modeling the heterogeneity within these cohorts. So how can we do better? So what I've talked to you today about today so far is effectively a case control type comparison, where we have a group of patients and a group of controls, and we aim to find differences between them. And I should say I'm not just limiting myself here to classifiers. The same logic applies to things like statistical parametric mapping, GWAS, as well as classifiers. So the standard thing to do, and there are many studies to this effect, is to say, well, we're not dealing with one clinical phenotype. What we might have is multiple different subtypes, for example, using, for example, clustering algorithms. And this is not a new idea. This lies really at the heart of initiatives like this one, like the ARDOC initiative, which effectively aims to take these diagnostic groups that are defined based on symptomatology by integrating data from many individuals. The idea is that we, biological data, symptom data, cognitive data, etc. Excuse me. We derive. We can apply some algorithms to get subtypes or clusters that are better reflecting the underlying biology. Now, this idea sounds really good, and I think initiatives like ARDOC have done a very good job in defining. This is the ARDOC matrix, which defines things like domains of functioning across multiple levels of analysis. So that kind of descriptive value is quite, it's been quite a nice contribution. But on the other hand, the idea of subtyping participants is not new at all. We reviewed the literature in this field a number of years ago. There are hundreds of studies aiming to do this, and there have, we still haven't converged on a set of meaningful subtypes for any disorder. And I think one of the reasons for this is this nice, neat framework doesn't really specify how we move from biological measures to subtypes, and this is really where the magic happens. So I'd like to talk about our approach for this. This is just one possible approach. There are others, but our approach sort of generalizes this notion that we're either talking about patients and controls or subgroups of patients, because those are obviously only one possibility. We could also think of things like a spectrum of functioning or an aggregation of many different causes in the same clinical label, and we'd like to be able to handle all of these. So our approach to that utilizes some of these large public population-based samples that are coming online. So we sample variation across lots of, tens of thousands of individuals. And it also leverages an innovation that we developed a number of years ago that generalizes the notion of pediatric growth charts. So this is a normative modeling approach that we've been working on quite a lot. All of you will be familiar with this notion where we plot some biological parameter, for example, weight or height, function of age, against centiles of variation within the population. Now, the idea in normative modeling is that we just substitute these variables for things that might be relevant for neuroimaging. So some measure of biology, for example, neuroimaging, as a function of some arbitrary covariates. So, for example, we could say we could aim to predict cortical thickness as a function of these demographic features at each location in the brain. Now, why does that? Is that what's purpose of doing that, so by doing that across many measures, we can build up this quantitative model of how individuals differ from one another, such that we can then make predictions, statistical predictions at the level of the individual. So then by doing that across cohorts of individuals, we can identify subjects that might have a somewhat atypical pattern, and we can use this as a basis for subsequent stratification without needing to assume that clusters exist in the data at all. So this is work from PhD student in my group, Sage Rutherford, and she aggregated a really large sample of public data sets to get a really good estimate of cortical thickness right across the lifespan. So there's more than 60 or around 60,000 individuals in this sample, and you can see that as we move across the lifespan, there are very different shapes in some of the variables that we consider, and we get really nice estimates of variability across the lifespan. Now, when we take these as features for a subsequent analysis, the simplest thing we could do is to do a case control comparison. And when we do that, by transforming the raw data to these population centiles, we get stronger case control effects across nearly every disorder, which is one benefit. But really, the real benefit is the ability to map individual differences, as I've alluded to. And to highlight this notion, I'm going to refer to work from Thomas Wolfers and Mariam Zabiti, both of which have moved on to postdocs elsewhere, and both have made really nice contributions to the field, I think. So across multiple different disorders, what I'm showing you on the left-hand side here are the classical group differences, and on the right-hand side, I'm showing you the individual level deviations from this model. So how each individual deviates from the expected pattern. And I'm showing you the overlap. So the regions that are coloured have overlap in more than, say, 2% of subjects, for example. So across schizophrenia, bipolar disorder, ADHD, and autism, we can see a consistent narrative emerging, is that we have in schizophrenia robust case control effects and very little overlap across individuals. Bipolar disorder, some effects and still very little overlap. And in ADHD, something similar. In autism, we have something slightly different occurring. So we have no case control effects, yet we have individualized effects across virtually every region that we could consider. So the idea is that this extreme inter-individual variability in autism is masking case control effects, and I'll show you how that works in a moment. And you might say, well, this is obvious. You could always do this, and this would be the case, and it's not true, because in dementia we get a very high, very consistent pattern of abnormality, or atypicality, if you like, in the medial temporal lobes, where up to 25% of individuals have things in the same region. So it provides us a way to quantify and map inter-individual variability in these disorders. So so that's quite nice. We can learn a lot about how individuals differ from one another, but we can also take it further towards the notion of precision medicine. We could take these deviation images, and we could say, for example, derive risk scores, and there's a method that Charlotte Prasa in my team is working on in this area. We could take them as we could try to find some kind of set of overlapping profiles for different individuals using matrix factorization techniques, or we could apply normative models as a basis for clustering, and this is the example that I'm going to show you now. So, as I've said before, so in autism, I should mention this is the EU-AIM study, which is one of the largest single studies of autism that's ever been conducted with neuroimaging measures. So it should, in theory, be quite well powered. Despite that, we see very few case control differences in autism. Yet, when we can take into account an assumed reference model, so we reframe everybody as in terms of centiles from an expected distribution, suddenly we see very widespread deviations in individuals with autism and not so many in controls, which is what you would expect. I should mention this is all derived out of sample as well. And what the way to interpret this is that there are very many differences in autism, but they're very idiosyncratic. So the same individual, different individuals don't have the same characteristic pattern. So if we could then try and unpack that a little bit and try to say, well, you know, what's going on in this cohort? And so if you were to train a clustering algorithm on the basis of these deviations, it turns out, I mean, I'm not going to say there are definitely 5, but we have, you know, 5 biotypes was preferred by this this model. But one thing that's immediately striking is you see that some of the individuals with autism have decreases in cortical thickness with respect to controls, and some have increases. So all of these things kind of cancel out one another, and that explains why we don't see any case control, or very few case control differences. Moreover, these biotypes map onto both symptoms and genetics more strongly than do the classic diagnostic labels. And so here I'm showing you the correlation across different symptom domains in autism, ADHD, and related disorders across the whole cohort. And you can see that when we consider variation within one of these biotypes, the associations get stronger and more of them become significant, despite a smaller sample size. And the same is true of the polygenic scores. This provides some indication that these things might be quite meaningful. Okay, so that's a little bit about techniques for understanding variation in clinical cohorts. I'd like to turn attention now to you know, what kind of features that we can derive to estimate these kind of models. How can we learn about the representations that might be occurring within brain structures that provide the ability to differentiate individuals from one another, and to make more accurate predictions of outcome or diagnosis. Okay, and I'm going to focus for the first part on methods for learning connection topographies. Now, what's a connection topography? A connection topography is a slowly varying pattern of connectivity that is preserved as it is projected from one region to another. So that means that that areas that are closely connected in one region are closely connected in another region that they may project to. And the most well-known example of a connection topography is the somatosensory or motor homunculus, which is represented in the primary motor or primary somatosensory strip. But in fact, it turns out that this is a fundamental organizing principle of brain function, and so it's found in fact in many regions. And Kuhn Haak, who's been a collaborator for a number of years, developed a very elegant method to be able to estimate these kind of connection topographies. And I won't go into it in detail, but it's based on a manifold learning technique that aims to, roughly speaking, look at the similarity of voxels within a region with the rest of the brain, construct some kind of similarity metric that we can then use to find these smoothly varying connectivities, they're called. We can do it from resting state fMRI. So I mentioned before that this is quite a fundamental feature of brain organization, and one that's particularly of interest to us is patterns of topographic connectivity in the striatum, because corticostriatal circuitry is really important in Goaldirected behaviors, and it's implicated in nearly every psychiatric disorder that you could mention. And this is a theoretical model for how these things are this connectopy is organized. It's derived from invasive tracing studies in non-human primates. It's never been, up until this point, it had never been shown in humans before. And the idea is that this is a cross section of the striatum, so the caudate, putamen, and globus pallidus, and the idea is that connectivity varies slowly as there's a gradient of connectivity across the striatum. So we used resting state fMRI from the Human Connectome project to estimate some of these striatal gradients, and these are the projections of these gradients to the rest of the brain. And you can see just visually that we're getting an excellent correspondence between what we can only theoretically infer exists in humans on the basis of invasive tracing studies with what we can estimate quantitatively using resting state fMRI. And it's pretty clear that those representations are really quite exquisite, I think. Moreover, it turns out that these things, these connectivity gradients, map onto goal-directed behaviors. So we can predict things like psychological well-being, delay discounting, that's a measure of reward and delay sensitivity, a relational task, and so on. So these things they're meaningful in the sense that they predict the behaviors that we think they ought to. Now, another, I think, really important feature of these connectivity gradients is that they can overlap with one another, which is a really important feature for brain organization because the brain is only so big, and we quite often see multiple representations occurring in the same brain regions. So just think of the visual cortex, for example. There are representations of things like polar angle as well as eccentricity in the same region. Similarly, in the striatum, there are multiple representations superimposed upon one another. Okay, and that's been shown again with tracing studies. This is work from Marianne Olderhinkle that was just published a couple of months ago in eLife. And so this is what I'm showing here is these overlapping representations. So this is the what we call the zero order connectivity mode, which effectively recapitulates the known anatomy. So you can see a smooth correspondence between the caudate, the putamen, and the ventral striatum, and so on. This is the first order connectivity mode. So this is the one that I've just shown you. So this recapitulates the work by Susanna Harbour and shows this gradual connectivity gradient that we can also observe in non-human primates. So these things are superimposed upon one another within the same structure. It turns out we can keep going, and this is the second order connectivity mode, which we can estimate again using data from the Human Connectome project. And what is very striking is that this third connectivity mode has a very clear spatial correspondence with measures of dopamine transporters. Sorry, this should be transporters, but and we can only get this using sort of invasive methods like PET or SPECT. So we can derive with non-invasively with resting state fMRI a proxy measure of dopamine availability in the striatum. Now, if you've studied psychiatric disorders, and you'll know that that's really crucial in many of is a really crucial locus in many psychiatric disorders. So these scatter plots at the bottom just quantify the strength of this relationship. So you can see the connectivity mode and that SPECT image are very strongly correlated with one another. Now, it turns out that these, and this is this correspondence that I've just been discussing. Now, it turns out within the Human Connectome project, there's different measures of, for example, alcohol and tobacco use, for example, and these things, measures that we derive from these connectivity gradients, are predictive of the amount of tobacco or alcohol use that these individuals might have have been using, basically. I'm going to show you also. So this is work from Peter Mulders, who's a psychiatrist with us at Radboud MC, and he's been working on a very interesting data set, which is referred to as the mindset data set. Now, this data set was designed to be realistic. So the way that subjects are recruited is that they're sampled effectively randomly from people that go through primary or secondary care within the psychiatry department. So it's very cross-diagnostic by design, and and comorbidity is embraced, and it's in fact the rule rather than the exception. So this is a very realistic perspective of what we might see in the wild, so to speak. And I'm just showing you here a Venn diagram that shows the number of diagnoses that each of these participants might have. So you can see that there are many more individuals within this cohort, remembering sampled randomly from people coming through the clinic, then there are only a subset of these individuals would have one diagnosis. So this again calls into question this notion of using clearly defined diagnostic entities that are distinct from one another, because that's not the clinical reality. So this sample is also deeply phenotyped. So we have many different instruments that assess many different aspects of cognitive function, many different symptom scales, and so on and so forth. So the first thing that Peter did was he performed a factor analysis on this panel of different measures, and he derived a set of domains of functioning. So things like negative valence, cognition, social functioning, and arousal or inhibition. He did it on a subset of these participants, and then just to be sure, we did it again in a replication sample. And you can see that the correspondence between these different domains is quite exceptional. And this is really a means to derive quantitatively in a real clinical population some of these different domains that might be specified in a theoretical framework, like, for example, ARDOC. So the next thing Peter did was he took a set of estimates of these corticostriatal connectopes, and he tested for associations between these domains. And it turns out that using a technique known as canonical correlation analysis, I'm not going to describe it in detail, he can detect an association between these domains whereas there was no association with diagnosis. So this really speaks to the need to shift away from binary yes or no decisions and really sort of embrace, on the one hand, heterogeneity in across different individuals, and also try to assess individuals across different cognitive domains. Okay, so I would also like to talk about learning some of these. So this is up till this point. I've been talking about learning a particular type of representation, a connectopic representation. But in many cases, we might be interested in learning something more general. And the archetypal example of this kind of notion is something like a deep neural network, where we have layers of abstraction as we sort of map from some kind of input, for example, an image, to a prediction. And these things, I mean, I don't have to tell you about the success that deep learning has had in many different domains over the past years. And so this is more along the notion of learning a representation for a particular task. And it turns out that deep neural networks have been applied quite successfully in many different domains of neuroimaging, for example, predicting age. And this is a particularly interesting example, I think, because this complex representation that we get from learning from a deep neural network, where we successively abstract, derive more and more abstract features to the point where we actually have a prediction, actually doesn't perform better than a linear model in this case. That's a topic for a different talk, but I'm not going to go into that today. But in some cases, we do get a benefit by employing these deep neural networks for prediction. For example, this is work by Han Peng that in fact they won a prediction challenge in 2019 using a model like this. The point is that when we learn these kinds of applying these kinds of deep learning models to neuroimaging data, what we end up with is a representation that's very finely tuned for a particular task. For example, for predicting age, which is fine if that's the only thing that we're interested in. But as neuroscientists, we often want to disentangle these representations and perhaps apply them to a different task than the one that they might have been trained upon. So this was another piece of work from Mariam Zavihi during her PhD, and the idea is that rather than learning a tuned representation for a particular task, we want to learn a general representation for human brain function that is interpretable and that we can control it in the sense that we can emphasize the aspects of the representation that are predictive of certain tasks. So for this purpose, we use the Human Connectome project data. It has a number of fMRI tasks which are designed to cover the range of functions that we might be interested in different cognitive functions basically. But we also sample the whole cerebral cortex using these different tasks. So we get a broad representation of cognitive function and an equally broad representation of the different brain regions that subserve that. So what Mariam did is she estimated what's known as an autoencoder. So that's a model a deep learning model that's trained to reconstruct images. And the idea is that we can take this central representation as a compressed code for the differences between the things that we're learning. Mariam used this is some technical details which are not particularly important. She used an autoencoder with a particular infrastructure, and we take this middle layer and apply again some kind of manifold learning technique to learn something about the structure for which this model has learned. Now, when we do that, you can see that this is by applying this manifold learning technique, we can see that we can learn the differences between the tasks. That's not surprising because they're quite salient from one another. And then we can reconstruct what the typical example of these things. So we can map back to the input space to see what this representation is actually showing us. One sort of problem, if you like, of this approach is it's unsupervised. So it learns the highest axis of variance in the data. So, for example, these clusters have very little relation between things like age and sex, which might be of interest if we want to be able to learn a particular representation that has some kind of balance between factors that we might be interested in, and also being able to accurately reflect the different domains that we're interested in. It turns out that this can separate the tasks quite well and performs better at reconstructing the data than a linear model, for example, PCA. What is desirable is, as I mentioned, is to be able to control this, to be able to constrain the latent representation to learn a particular aspect, cause it to focus on a particular cognitive domain, for example. For this purpose, we trained an autoencoder that, on the one hand, reconstructs the data, but also enables us to predict demographic variables accurately from this latent representation. So when we do that, we get still a separation of the tasks, but also we can then learn something of a gradient that describes how people differ with respect to these demographic features that we train it on. By transferring it to UK Biobank, so we take the Human Connectome project data, which has approximately 1,000 subjects, apply it to UK Biobank, retrain it a little bit so that we can learn the different features of the different data sets. And UK Biobank, of course, has a very large number of subjects, tens of thousands. And this is the representation that we get. So when we don't employ this semi-supervised feature, we get some kind of representation that does reconstruction, but it doesn't necessarily relate to any of the other variables. Whereas if we inject that into the learning function, we can then derive a representation that is a compact summary of how subjects differ with respect to one another, with respect to the demographic features that we put in. What we can see is that we learn, roughly speaking, one dimension that describes age-related variation, another that describes sex-related variation, and we can apply this then in a normative modeling framework to get centiles of variation and subsequent predictions, as I've shown you in the first part. It turns out that when we do that, we get much stronger associations between clinical variables. So in UK Biobank, we have a whole bunch of what are called NIDPs, or in other words, non-imaging derived phenotypes. They're demographic features, they are clinical variables, they are things like physical variables, blood pressure, lots of different lifestyle factors, and so on and so forth. And when we apply this model with respect to an unsupervised model where we see no associations within the latent representation, here we see much more strong associations, which gives us an indication that the representation we've learned maps much more closely onto clinical demographic features. We can then, again, as I've shown you in the first part of the talk, take this method forward towards providing single subject level inferences, novelty detection, normative modeling, and so on and so forth. Okay, for the last part of the talk, I'd like to look a little bit to the future. And this is work that we are just starting on, in fact. And the idea is that I've shown you a lot about how we can use biological measures to get biomarkers, separate groups, to stratify cohorts, and so on. But of course, the brain doesn't live in a jar, and we are embodied beings, and we live in an environment, and we have, we behave and we act, and all of those things, ideally, to get an integrated perspective of each individual, it's nice to measure. And so two technologies that we're exploring at this time for doing that is, on the one hand, digital phenotyping, so deriving quantitative measures of behavior from smartphones. And then we can also quantify environmental variables using satellite monitoring technology. And I'll talk about those in a moment. So this is a project that we're just starting. And the idea is we have a number of platforms. We use two different platforms for this. So these are what's called BioAffect and BHAP, and they're basically passive monitoring platforms. One monitors typing dynamics, that is how quickly and how many mistakes you make when you're typing, not explicitly no content. And the other, the BHAP platform, measures things like how often you go out, how many times you make social interactions, how many phone calls you make, again, anonymized with respect to who they are, or things like that. So it protects patient individual privacy. And the idea is we get time series with these measures that are generated automatically in the background. So this gives us a quantitative measure of behavior in the real world. So it's quite different from the measure of behavior you might get in an experimental setting or in the clinician's consulting room. And the idea in this project is that we use these different measures, these sensor level data. So we get really long, really dense time series from these individuals. We estimate a set of latent behavioral time courses from which we can say, generate summary statistics. So, for example, how long is each individual staying in a particular state? Sitting at home versus going out socializing. What are the circadian rhythms doing? Are they waking up early? Are they going to bed late? Those kinds of things, and use those as predictors of mental health in a real-time, real-world setting. And the idea is that we'll integrate these with measures of biology derived from neuroimaging along the lines I've shown you already. I don't have any results to show you at this point. We've got a couple of PhD students just starting on these projects, but I hope maybe next time, if I'm invited back, I might be able to tell you more about what's happening in this project. The other aspect that I've introduced is how can we measure behavior, measure environmental influences in a quantitative fashion. So this is an example. This is work done together with Gunther Schumann and other collaborators in China, where we aim to measure different environmental features. For example, urbanicity. So urbanicity in the sense of, how long do individuals live in cities? Have they spent their whole lives there? Have they moved to the city? How built up is it around them? All of those things are potential predictors of mental health. So we can measure that quantitatively using satellite data. And there's a number of different proxy measures that we can get. So, for example, nighttime light, how much light pollution is there? Vegetation indices, how many trees, and what's the ground cover like, and so on and so forth. And we have data for those different measures derived from a number of different satellite systems in China and in Europe. And so on the basis of these measures, we can we derived a proxy for urbanicity, and we can, because we have very fine-grained information about how these measures change over time as areas are built up, where people lived at certain times. All we would need in principle to make a prediction for an individual is the spatial location at which they lived at certain points in their lives. So in a number of cohorts, this is the Imogen cohort, and this is Chimgen cohort. We have that information, so we can determine quantitatively not only how urban is the subject's current environment, but how at what times in the past have they lived in an urban environment, and how has that changed over time. Now, it turns out that these measures of urbanicity are predictive of brain structure, brain function, and also in a time-dependent manner. So you can see that there's a gradient between people that have, this histogram at the bottom shows people that have always lived in the city versus people that have moved to the city before 14 years and have moved to the city after 14 years. And you can see that in many areas, there's a gradient in terms of how these measures of urbanicity affect brain function. And these also predict mental health as well. So we can use these measures to predict sensitive periods. So if this schematic is showing how much additional risk you might confer for getting a psychiatric disorder or having mental health problems, depending on when you move to the city. And you can see in this case there are approximately 2 peaks which map onto different brain mechanisms. And I'll refer you to the paper which is published quite recently in Nature Human Behavior for further details about that. This is kind of like the seed for a new project that we'll be starting in the coming months, and this is a Horizon Europe project that aims to really go the whole way in integrating satellite data with neurobiology, with genetics. And there's a seminar series which will be taking place in the coming weeks. I'll warmly invite you to attend that, where we'll outline the strategy and so on and so forth, how we will tackle these challenges. So just to wrap up, I'd like to leave you with a couple of take-home messages. So in the first part, I'd like to convince you, I hope I've convinced you that this very strong over-reliance, I would say, on case control thinking has really hampered us in terms of finding biomarkers that predict outcome or enable us to stratify groups. I think in the second part, I tried to show you that, in order to get the optimal differentiation between individuals, how that maps onto behavior and so on and so forth, it's important to be able to learn the right representation. And I showed you 2 examples of how to do that. So this conic topic mapping approach, and also learning generalized neural representations using neural networks. And in the last part, I've just given you a taste of the things that we'll be doing in the future to integrate multiple aspects of technology to really get an integrated perspective of all of the things that contribute to mental health and well-being in a quantitative fashion. I'd just like to thank lots of people that have contributed to the work that I've shown you. Of course, I'm just here taking the credit for it, but it's the work of many people. I'd like to acknowledge that. Just also to say, if you're interested in the kind of stuff that we're doing, please get in contact. There's lots of interesting methods development work going on, and sort of the applied stuff as well. And I'd just like to thank finally yourselves for your attention.
They said, to let people connect, even though they know that people should.
Thank you, Andre. It was super interesting. Can you hear me? You have some noise. Maybe it's noisy here. Does anyone have some questions? Do you want to share it in the? Please share it in the chat box if you can. Wait, I can. Okay, yes, I can see some questions. So I didn't see who was first. Victor. Okay, Victor, go.
Hi, thank you for the great talk. It was really interesting. And well, I mean, I'm quite like all the aspects of machine learning and this kind of thing about to try to decipher the different groups in the population and try to do prediction, to better the prediction that we can have with biological data. But that also, so I have like two questions. It's more about the technical part. Sorry for those who are not interested in this part, but it's about how do you plan to tackle the curse of high dimensionality that you face when you have the wealth of data that we have nowadays, either in the neuroimaging data that you have, but also combining all this data, even combining maybe genetic data and this kind of thing to produce some kind of model or prediction that is reliable. Because when you have that much data, how do you do? Because it's you've got in the end more data points in more dimensions than actual participants in your data set.
Well, it's a very good question, and I would say bringing domain knowledge is really important in that respect. So fitting an unconstrained model that can find any kind of association is probably too flexible. So finding the right representation for the data on the one hand, building what we know about brain organization and adapting the models for the complexity of the data. So for example, if you're in machine learning, you're working for Google, your task is image classification. You have millions of examples that are 2D image patches to train your models on, and we don't have that. We work with 3D data largely, and even with UK Biobank, we only have a fraction of that data. So constraining the complexity of the models by design is one aspect. Injecting domain knowledge to make sure that we're using what we know about the brain to constrain the representations that we learn. And validation. So if we have, how do we show that these things that we've learned are meaningful? And that's the most difficult, and I would say the most crucial step. So we can relate them to genetics, we can relate these representations to behavior, we can relate them to what happens in the future. And all of those things, I would say, add up to giving plausibility to the models that we learn.
All right. Thank you. And just the other question that I had is that I saw that you were working with a lot of different data sets. How do you take into account the heterogeneity of the different biases that you can have with each data set? Because even if they are all in MNI space or something like that, they will have their own bias, each one different.
I have a whole talk about that.
Oh,
so basically, in short, we design models to handle that. So in a linear modeling context, we have, for example, a hierarchical model that accommodates the different biases, learns the side effects, and aims to generalize them across different data sets. If you are doing it in something like a deep learning context, you would have to do it differently. And we could do things like transfer learning to say, we'll train on this site, then we learn the site effects as we apply it to a new data set. But it's indeed an important point. I mean, if you want, I can send you some references about this kind of stuff that we've done in that domain. But there is a lot, that a lot of our effort goes into dealing with those kinds of problems.
Okay, thank you.
Thank you. Steph, do you have a question? You were the second.
Yes, thank you. Thanks for the talk, Andre. Very exciting. I was particularly intrigued by the dopamine receptor distribution that you showed with fMRI in comparison to PET imaging. And now obviously PET imaging is quite invasive, whereas fMRI should not be. And I was wondering how far we are away from the clinic. Like, is this something that can be readily implemented, or do we still look at the classical 10 years down the line?
No, it's so there's a project going on at the Dundas actually to do that in Parkinson's disease. I didn't show you any results with Parkinson's disease, but it turns out that those things can predict not only the magnitude of symptoms like tremor, for example, but also the hemisphere. So if you so there is for things like sample enrichment, where it's a lower bar for clinical validation than perhaps predicting outcome, I think they're closer than any other neuroimaging derived biomarker that I could name. But they're still realistically, a number of years away. The clinical validation process takes a very long time, but there are steps in that direction already.
Thank you.
Valentina, you have a question?
Yes.
Okay, go ahead.
Thank you, Andrew, for your nice talk. I had a question on the last part that you presented. I was wondering, how did you measure mental health in this big sample that you...
Well, you can say, I mean, there are different, I mean, it depends which part you're talking about, but usually it could be something like reported symptom self-report symptom levels, questionnaires, incidence, number of diagnoses, or a number of sort of interactions with health services and things like that.
I was talking about the last part where you showed the plots with the world map and the mental health correlated with living in the city or
so it would be things like so if you have multiple sort of time points, you have some, for example, symptom scores that subjects are filled in at the end. So they're recruited from existing clinical studies.
Okay. So you have like variety of clinical testing.
Different clinical instruments, for example.
Okay. And then you computed an index of mental health, or you went true to the variability of your data?
Well, first indices. I mean, it's quite a preliminary. It's quite a preliminary sort of report in that sense. I mean, there's a lot of work that remains to be done in terms of predicting. I mean, I would say that what we've shown at the moment is an association level.
Okay.
It's not. We're not sort of getting down to the level of statistical subject level prediction at this point.
Awesome. Thank you so much.
So I also have a question. I was thinking about the diagram that you show where the different diagnoses have an overlap, but also some differences between them. And also with all this data, we really now gain a lot of information from the environment, also real time. So can we also think about new diagnoses, new conditions? So can we actually not just train the data, but learn from the data from all these new frameworks that we are exploring, and we have all this power to understand the latent representations?
Yes, we can. It's a very slow process, though. So the and there is still a lot of, I mean, it's in many domains, it's hard to, I mean, symptoms, many illnesses are still treated at the symptomatic level. So there's a lot of our infrastructure is set up on that, on that framework. In some domains, diagnoses are less important than others. And, most, I would say good clinicians would try to improve quality of life and functioning and things like that. And diagnosis, perhaps as a means to enable that to happen. What I would like to see in the future is more sort of empowerment in terms of things like monitoring systems. So if we have a smartphone-based monitoring tool, then, people can sort of get early warning signals when things are starting, perhaps, the behavior, some sort of signature in the behavioral time series is starting to, indicate perhaps the first parts of a relapse into some, particular pattern of behavior. And being able to identify that early, that opens up all kinds of different avenues.
Yes, also so longitudinally, like early onset kind of alarm behaviors and then like the treatment will be like with like pharmacological treatment or psychological treatment. How this once we know
I'm not
how
I'm not a clinician really, so I wouldn't sort of like to comment much about that. But I mean, the idea is, if we can sort of have a more integrated perspective of what's happening in a real-time basis, then that's more information that we can use to make better decisions.
Does anyone has other questions? I was thinking also about, because you also talk about a little bit of genetics, how also can we see similarities between hereditability of some behaviors or pathologies. This is like a super big data set that now we can model.
Well, I mean, in a certain way, we have another very rich source of information in the genetics domain, and understanding the heterogeneity in that domain has similar and different challenges. So we have, an even higher dimensional phenotype. If we get down to whole genome sequencing, then we have an even richer set of sort of measures to consider, and then dealing with heritability, population stratification, all of those kinds of things, are quite big challenges as well. And take, for example, autism. There are many genes associated with, those different, with autism, for example, and they may have different pathways that converge on similar or different things. So it's a very rich space, but there's not a lot remains to be done. That's clear.
Yeah, I agree. But yeah, super interesting that now we can monitor and modeling for this. I cannot see other questions. I don't know if we have questions on YouTube. If you have other curiosities, something to please make or write it or let us know. Okay, if there are no other questions, maybe we can thanks again, Andre, for the presentation. It was super interesting, and we really look forward to the next results that you just show us the rationale of the studies. It was super interesting, and we are really curious about it. Remind you our weekly Neurocino paper discussion that you can join or rewatch them online. And I hope everyone a nice evening or morning, depends on which part of the world you are. And thank you again.
Thank you. Bye.